Here's the essay. Introduction. The scientific landscape of molecular biology underwent a dramatic transformation during the late 1950s and early 1960s. A central enigma, the mechanism of protein synthesis, demanded a deeper understanding. The question of how the genetic code, encoded within deoxyribonucleic acid, or DNA, dictated the production of proteins represented an extraordinary challenge. This essay undertakes a thorough evaluation of the groundbreaking work of 1960. 1. Dependence of cell-free protein synthesis in E. coli upon naturally occurring or synthetic polyribonucleotides, by Marshall Warren Nirenberg and Johann Heinrich Matthaei, a seminal contribution that profoundly reshaped our understanding of this complex process. This work presented the first direct experimental evidence supporting the idea of a coding system for protein synthesis, a critical finding that marked a pivotal moment in the history of genetics. The following analysis proceeds section by section, meticulously examining the claims, methodologies, and implications of this landmark publication. Section 1: Introduction and Rationale. The introductory section of Nirenberg and Matthaei's paper artfully lays the groundwork for their investigation. The authors begin by acknowledging the established, though poorly understood, role of ribonucleic acid, or RNA, in protein synthesis. They emphasize the limitations of existing knowledge and, specifically, the lack of unequivocal evidence supporting a direct link between the genetic code and the ordering of amino acids within a protein. This section provides a clear rationale for their experiments, highlighting the need to investigate the relationship between RNA and protein synthesis in a cell-free system. The authors underscore the promise of this approach, suggesting that the absence of the complex cellular environment would allow for a more direct study of the underlying mechanisms. They point out the possibility of using synthetic RNA molecules of known composition to decipher the genetic code. A key aspect of their argument involves the advantage of eliminating cellular complexities, allowing focus on the fundamental biochemical interactions. This section, therefore, functions as a precise map, guiding the reader through the logic that underpins their experimental design and offering a clear justification for its innovative nature. Section 2: Materials and Methods. Cell-free system establishment. The description of the materials and methods reveals the ingenuity underlying the experimental approach. Nirenberg and Matthaei describe the preparation of a cell-free system derived from Escherichia coli, or E. coli, bacteria. A critical component for their work, the authors carefully outline the steps involved in preparing the bacterial extracts, emphasizing the importance of obtaining ribosomes, the cellular machinery responsible for protein synthesis. They detail the solutions, the purification steps, and the precise incubation conditions employed to ensure the production of proteins. The methods section further describes the use of radioactive amino acids, particularly carbon-14 labeled amino acids, to monitor nascent protein synthesis. This careful labeling permitted the tracking of amino acid incorporation into the synthesized proteins. The authors also detail the preparation of the RNA templates, including naturally occurring RNA isolates and the crucial synthesis of synthetic polyribonucleotides. This detailed description of methods reflects a commitment to rigorous experimental control and reproducibility, key pillars of scientific advancement. The careful description of the cell-free system, the source of bacterial extracts, and the methods used for synthesizing proteins are all critical for understanding the subsequent findings. Section 3: Synthetic polynucleotide incorporation into polypeptides. The core of the paper resides in the description of the experiments. Nirenberg and Matthaei describe the addition of synthetic polyribonucleotides to the cell-free system. The groundbreaking finding was that the addition of synthetic polyribonucleotides, specifically polyuridylic acid, resulted in the incorporation of amino acids into polypeptides. Their observations demonstrate that, in the presence of polyuridylic acid, only the amino acid phenylalanine was incorporated into the polypeptide chains. This observation represented a truly defining moment. A synthetic RNA of known composition, in this case polyuridylic acid, acted as the template for protein synthesis and determined the amino acid sequence, the ability of a simple synthetic polyribonucleotide to direct the incorporation of a specific amino acid suggested a fundamental mechanism. The authors presented compelling evidence showing that the presence of the synthetic polymer was essential for the observed incorporation. The control experiments, designed with a meticulous attention to detail, demonstrated a clear correlation between the presence of synthetic polyribonucleotides and the production of the polypeptide. This section provides the essential experimental data that initiated the deciphering of the genetic code. Section 4 Further characterization of the synthetic polynucleotide-directed polypeptide synthesis. The ensuing sections of the paper delve further into the nature of the protein synthesis directed by synthetic polynucleotides. The authors examined the characteristics of the newly formed polypeptides. They addressed the question of whether the incorporation of amino acids was in fact attributable to the directed chain reactions. Critical to their claims involved a consideration of the stability of the synthetic polymers in cell-free systems and any limitations that might impact their interactions. The authors explored the size and composition of the polypeptides. They discussed the effect of the concentration of the synthetic RNA molecules on the rate of amino acid incorporation. They further considered the impact of variations in the experimental conditions, such as temperature and the concentration of magnesium ions, on the process. The results of these additional experiments all consistently supported the central conclusion that the synthetic polynucleotides were intimately involved in the process of polypeptide chain synthesis. The careful analyses that accompanied these additional experiments further strengthened the author's claims. Section 5 Evidence for the codon-uracil-phenylalanine relationship. The conclusive part of the paper presented the culmination of their research. The authors interpreted their results as evidence for a specific relationship between the polyuridylic acid and the amino acid phenylalanine. They proposed that a sequence of uridines, a codon, corresponded to the amino acid phenylalanine, a proposal that paved the way for the understanding of the genetic code. Using their experimental data, they proposed that a triplet of uracil bases in the messenger RNA, or mRNA, coded for the amino acid phenylalanine. This conclusion stemmed directly from their observations regarding the amino acid incorporation in the presence of polyuridylic acid. The authors acknowledged the limitations of their work, recognizing that their experiment provided evidence for only one codon. They clearly stated their interpretation as an initial key to the coding system. They also discussed their findings that the initial results were not in disagreement with some of the theoretical frameworks that had been proposed earlier. This careful approach to interpreting the results, acknowledging both the strengths and the limitations, showcased an example of scientific rigor. This critical section, therefore, marked the beginning of the effort to decode the genetic language. Section 6 Discussion of the implications and future directions. Nirenberg and Matthaei's concluding section. The discussion presents a thoughtful assessment of their findings and their implications. They recognized the significance of their work, highlighting its importance in the context of ongoing investigations into the genetic code. The authors discussed the possibility of using other synthetic polyribonucleotides to further elucidate the code, suggesting that by using different polymers, they could identify the specific codons that corresponded to other amino acids. They also discussed the implications of their findings for the study of other biological systems, including viruses and higher organisms, and outlined the pathway to understanding the language of life. They envisioned a future where the genetic code could be fully deciphered, opening new avenues for the exploration of biological processes. The authors recognized their ability to offer a crucial starting point. The potential for future research lay in the use of new synthetic RNA molecules. This discussion, therefore, laid the foundation for the explosive growth of molecular biology that followed. Conclusion: the 1961 publication by Nirenberg and Matthaei constitutes a landmark achievement in the history of molecular biology. Their systematic and insightful experiments provided the first conclusive evidence of a direct relationship between RNA sequence and protein synthesis, by demonstrating that synthetic polynucleotides could direct the incorporation of specific amino acids into polypeptides in a cell-free system. They initiated the deciphering of the genetic code. The work's meticulous methodology and careful interpretation of results established a standard for the application of experimental rigor. Their findings offered a revolutionary framework for understanding the fundamental processes of life. The paper's introduction, methods detailed, and data presentation have had lasting impact on a wide array of fields. The authors' recognition of the limitations of their study and their foresight in suggesting future research directions showcase their deep understanding of the complex scientific landscape. The core discovery that polyuridylic acid directed the production of a polypeptide containing only phenylalanine was a crucial discovery. The legacy of Nirenberg and Matthaei's work is far-reaching. It paved the way for the complete elucidation of the genetic code, which, in turn, revolutionized fields such as genetics, medicine, and biotechnology. The impact extends beyond the immediate scientific implications. The paper serves as an exemplar of the transformative quality of scientific inquiry. The work stands as a testament to the power of careful observation, rigorous experimentation, and brilliant interpretation. It truly merits its place as a cornerstone of modern molecular biology.